Hello students, hello friends. Welcome to the fourth lecture of microbial cell culture and its applications. Hope all of you are staying safe and healthy. In the last three lectures, what we have seen is that we have seen why biotechnology is needed, how it can actually replace the petrochemical industry and what are the products it can produce. Then we have seen that what kind of what is a cell culture is, what kind of medium it is required, what kind of nutrient a cell requires for growing up. For instance, it require a carbon source, nitrogen source, anti-foaming agents, trace metals, cofactors and all of those kind of things. Then we also had a look that what kind of equipments for growing the cell culture, what kind of equipments for growing cell culture is required. So, we had a look at incubator shakers as well as the fermenters, and we have also seen what kind of fermenters are, we have also seen that what kind of fermenters are there, small scale, large scale, and we have seen that there can be different kind of fermenters, packed bed ones, but commonly used is in detail we have studied the stirred tank reactor, and then we also seen that how is the kinetics and the growth of microorganisms takes place. We have seen what is the different mechanism of cell division for yeast, fungi as well as bacteria. Then we have seen that what kind of growth phases there are, or what kind of bioreactor techniques like batch bioreactor, fed batch or a continuous system is there. We have also studied chemostat, and then we have studied also how actually to do the cell measurement, dry weight basis, spectrophotometer and all of those kind of things that we have studied. Then we have also studied and did the derivation how to calculate the doubling time with the biomass. So, in today's lecture, what we are going to do is we are going to focus on growth kinetics and specific growth rate using cell numbers. I would like to point out here is that cell number and cell masses are two different concepts. Cell numbers is total number of cells that is present in a reactor, and cell number concentration would become cell number divided by unit volume, while cell mass is total amount of biomass in the reactor or in the given volume. And when we talk about its concentration, biomass concentration, it becomes cell mass divided by per unit volume, and this cell mass we basically say typically dry wet cell mass. Okay. In the case of bacteria, it is still okay to use this term interchangeability, okay, because if all the cells are of same type, then you do not have a problem. But in the case of fungus, the use of this term in interchanging these terms can be little tricky. Okay. So, what we are going to do is we are going to focus on growth kinetics and a specific growth rate using cell numbers, and after that we have a look at scale up. Okay. What are the challenges in scale up, how you do the scale up, and what are the steps in between. Okay. So, in the last class, if you see, what we have seen is that when the microorganism is dividing, then basically to define the characteristic of the kinetics we use generation time or doubling time. Okay. Why it is important? If you remember, we have studied this, that why it is important. We know that a bioreactor that is producing fast and early is useful. We know that the old saying that time is money, it is very true, and it is literally true in the case of microorganisms, and not only in the case of bioreactor or microorganism or the bioproduct formation, it is also true for most of the industrial processes. If you remember, in the last class I gave you an example of auto industry, that how a 5-minute or 10-minute holding time leads to cost on the side of the manufacturer. Okay. So, one more point that needs to be emphasized before we move on to the cell number is that the growing and the doubling we are doing, this is specifically linked only to the log phase or the exponential phase. So, I think you have studied this many times, but I will just like to repeat it one more time. Okay. So, this is time, this is cell concentration, and it goes like this. This is our lag phase, this is our exponential phase, this is our stationary phase, and this is our death phase. Okay. And when we talk about doubling time, we are talking only specifically about this thing. Okay. Log phase or exponential phase. This is also known as log phase. I think you remember this and have studied this a lot of time. Okay. So, we have seen that when we want to measure the growth of the cell, we can use two methods. One is biomass and cell numbers. We have already done the calculation for the biomass, that how essentially the biomass is measured, and then how can we calculate the doubling time. Let us see how the cell number works. Okay. So, in this case, what happens is that we know that at the start there would be some amount of cell. Let us call that as N0. Okay. And then we will do the example, and this is the example that you will see. So, I will again use the same thing that if you remember we did it last time. Let us say we have an x1 at t which is equal to Td, which is t doubling time. X1 becomes x plus x, and then t is equal to t doubling, x becomes x plus x plus x plus x. So, let us try to do this numerically now. So, basically if you have number of cells as x or x number of cells, let us say x is just one number of cell. So, this is 1. After t equal to Td, that is doubling time, this x becomes 2. Okay. Again t equal to t doubling time, x becomes 4, and then let us say again t is equal to t doubling time, x becomes 8, right, because 1 becomes 1 plus 1 becomes, this one would become 1 plus 1, and this one would again become 1 plus 1. Now, this one again would become 1 plus 1, this one would become 1 plus 1, this one would become 1 plus 1, and this one would become 1 plus 1. So, what it is? 1, 2, 4 and 1, 2, 3, 4, 5, 6, 7, 8, right. So, 1 after t doubling time 2, after t is equal to t doubling time 4, and after t is equal to t doubling time it is 8. So, this is 8 here. Okay. So, how will it look? Well, you see here, if you see, follow my mouse, the number of cells 1 after 1 doubling time it is 2, after another 2 doubling time or 2 generation or 2 number of cells division it is 4, after 3 doubling time or 3 cell division it is 8, and after n doubling time it becomes 2 raised to power n. Okay. How it becomes? I can show it to you here. See. So, if you already agree in the beginning, if we have 1, this becomes 2, this becomes 4, this becomes 8. Okay. This I can, so here if that happens when the number of cells is 1, but if this number of cell here instead of 1, this is let us say is 3, okay, then how many of 3 would be here? 6. Okay. How many of the 6 would divide? So, we can do it like this, 1, 1, 1, this becomes 1 plus 1, 1 plus 1, 1 plus 1. So, how many it is? 1, 2, 3, 4, 5, 6. Now, this one would go, and so this one would go and make 1 plus 1, and this one would make another 1 plus 1. This one would make 1 plus 1, and this one would again make 1 plus 1. This one would make 1 plus 1, and this one would again make 1 plus 1. So, how many it is? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So, basically 12. Okay. Next will be 24. Okay. So, can I write it like this? This is 3. Okay. This is 3 into 2. This is 3 into 2 into 2. Okay. And this is 3 into 2 into 2 into 2. You see, after one cell division, after second cell division, and after third cell division. So, or I can write it like this: 3 into 2 raised to power 1. After second cell division, it is 3 into 2 raised to power 2. After third cell division, it becomes 3 into 2 raised to power 3. Okay. So, this is how exactly what we have done here. You can see this when we have number of cells equal to, if you follow my mouse, number of cells equal to 1 at 0 cell division or n 0 at 0 cell division. When after one division the number of cells becomes 2, or n 0 into 2, or n 0 into 2 raised to power number of cell division. So, that is 1. Here after 2 cell divisions, the number of cells become n 0 into 2 into 2, or n 0 2 raised to power number of cells division, which is 2 over here. Then if you look here, after 3 cell divisions, the number of cells is 8, or if the starting number of cell is n 0, then it becomes n 0 into 2 into 2 into 2, or you can write it n 0 2 raised to power number of cell division, which is 3 over here. If you have n number of cell divisions, then basically this is n into 2 raised to power n, okay, or n 0 into 2 into 2 into 2 into 2 n times. Okay. So, what it gives us? What it gives us is n t, the number of cells at time t, is equal to n 0 into 2 raised to power n, okay, where n is the number of cell divisions, n 0 is the initial cell number, and n t, as you can easily guess, is the cell number at time t, right. Let us have a look at it again. N t is equal to n 0 2 raised to power n. What is n t? N t is number of cells at time t. What is n 0? N 0 is initial number of cells, and what is n? N is number of cell division that has taken place. Okay. So, with this, basically if we know how many number of cell division has taken place essentially, or if we know a relationship between, if we basically know number of cells division that has taken place, we can calculate it, we can calculate T D, okay. That is the doubling time of the cells, right. So, we know that n t is equal to n 0 2 raised to power n. If I take log of this, it will become log of n t is equal to log n 0 plus n log 2, or log n t minus log of n 0 is equal to n log 2. So, what is n? N is equal to log n t minus log n 0 divided by log of 2. Okay. So, n is equal to 3.3 log of n t minus log of n 0. Okay. Basically, I just put the value of, so 1 upon log 2 is equal to 3.3. That is what I have just put in over here. Okay. Log 2 is approximately 0.3 something, 0.3, 0.393, something like this. Okay. So, 1 upon log 2 comes out to be 3.3. Okay. So, what we have seen? What we have seen is n, that is a cell division time, is log n t minus log of n 0 divided multiplied by 3.3. Okay. What is interesting here is that what is log 0? Sorry, what is log n 0 or what is n 0? As I said, it is an initial cell number, but if you remember how I told you about plating or inoculum in the reactor, that is what it is. That is what our n 0 is. Okay. And then if we know that the n, okay, so what is our then doubling time? Doubling time is simply T d is equal to T by n. What is T here? Okay. The T here is total growth time and n is, as you can see, a number of divisions. Okay. So, T d is total growth time divided by number of divisions or T by n. Okay. Let us have a look at an example and this will become little bit more clear. So, just to repeat, n is equal to log n t minus log n 0 into 3.3 and T d is T by n. Okay. Now, let us look at this example. The example is calculate the generation time or doubling time or T d of a bacterial population in which the number of bacteria increased from 10 raised to power 4 per milliliter to 10 raised to power 7 per milliliter during 4 hours of exponential growth. Okay. So, what has happened is this, that this is 4 hours, okay, incubation, and this is after the microorganisms population has increased. The initial population is 10 raised to power 4 per milliliter and this is 10 raised to power 7 per milliliter. Okay. So, what is our N t here? N t is 10 raised to power 7. What is our N 0? Which is 10 raised to power 4. Okay. And what is our T? T is 4 hours. Okay. So, we know the formula, N is equal to 3.3 into log N t minus log N 0, right. So, 10 raised to power 4 increased to 10 raised to power 7 in 4 hours, n equal to 3.3 into log of 10 raised to power 7 minus log of 10 raised to power 4. This comes out to be 3.3 into what is log of 10 raised to power, that is 7 and 4 is 4. So, 3.3 into 3. So, 9.9 or approximately 10. So, n is number of generation that has taken place is 10. Okay. Now, our time is 4 hours. Okay. This is equal to 240 minutes. Okay. So, our doubling time T d is 240 divided by n. So, T divided by n, that is 10 equal to 24 minutes, or if you want to do it like this, our T time, which is time of growth, is 4 hours, and n we have just calculated is 10, then your T doubling time becomes 4 divided by 10 equal to 0.4, and if you multiply this with 60, so 0.4 hours, or 0.4 into 60 is 24 minutes. Okay. So, this is how you can calculate the doubling time if you know the time taken for the growth and number of cells, and then basically based on that you can find out the number of cell divisions, and once that has taken place, then you can find the doubling time. Okay. So, these are the two ways essentially, one is the biomass, how we calculated the doubling time, uh, or using the number of cells. Okay. That is how you can basically calculate the doubling time. Okay. Now, let us look at the scale up of the microbial processes. Okay. One of the thing is that everything that we do at the laboratory scale, it eventually has to scale up. Okay. If we do not do the scale up, essentially we cannot produce the bioproduct in the mass quantity, which is what is required, and there are several challenges when it comes to the scale up. Okay. The challenges are not very simple to overcome. It require a dedicated effort, and it require a lot of experimentation before you reach the scaling up limits. Okay. So, before running 1,000 or 2,000 or 5,000 liter reactor, you have to have several steps in between. Okay. It is not possible to run a 0.5 liter or 1 liter reactor or do the shake flask experiments and then immediately run the bioreactor. Okay. Because there are a lot of things that needs to be, lot of decision that needs to take place. Like for example, if you have run a 0.5 liter bioreactor and now you have to run a 1000 liter bioreactor. Okay. It does not mean that whatever dimension. So, the first question would be, what is the dimension of the reactor that you need? Okay. What will be the diameter? What will be the length of the reactor? How will be the mixing will take place? How many impellers would you need? What would be the shape of the impellers? What is the RPM that you need to run in? Okay. What is the airflow that will be required? Okay. So, if you are putting one impeller in a 0.5 liter reactor, do you also need only one impeller, okay, in a 1000 liter reactor, or do you need 3 impellers? If you need only, let us say you need 3 impeller, then what will be the size of those impeller? Will the size would be where the increase in the ratio of the diameter of a small reactor or and the large reactor, or the length of the small reactor or the large reactor? What would be the airflow rate? Will the airflow rate remain the same? Will it increase? Should it increase? Should it decrease? So, there are several such questions that comes into the place before you do the scaling up, okay, and this needs to be answered. Then there is another challenge, then which is coming from the economics point of view. That challenge is, it requires a large amount of investment. Okay. Buying a 0.5 liter reactor is cheap, but buying a 10,000 liter reactor is expensive. Okay. And once you have bought that reactor, you need to make sure that your process would work in that, because if it is not working, that reactor cannot pay itself back, and hence that is a dead, there is a bad debt on the company. So, you have an inventory in the company, or you have a machine in the company that is not useful, and that is not useful in producing the process or producing the product. Okay. So, essentially essentially there you have to, whenever you decide to scale up, you have to really think very carefully and do a lot of experiments and calculation and extrapolation to come up with numbers that are feasible and that will lead to a successful process. Okay. Another challenge that comes is that when you scale up, you might not end up with the product that you have got in the beginning at the smaller scale. So, the product amount, the product type will be the same, but the quality of the product would deteriorate, or the quantity that you were expecting to produce, it will not be the same. Okay. So, that would mean that you were calculating everything, your profits, your production, everything at a certain place or at a certain rate. Okay. And but at the end of the day, the amount that you are producing or your manufacturing capacity is just the half. Okay. So, that problems you have to ensure that such a problem you do not meet. Okay. So, why? So, so the very important question is why in the scaling up such quality standard may not be met, and why cannot we just scale it up simply. Okay. The reason is because of the sentence that is just written below. The challenges are due to the mass transfer limitations and handling of large volumes. Because of this, there is a substantial challenge of scaling up. What do I mean by mass transfer limitations? As I have also explained earlier, uh, that in the scale up process, when we are saying that oxygen is homogeneously available or substrate is homogeneously available, this may not be true. For instance, just to give you a very quick and easy example, if you have 10 milliliter and you add 1 drop of dye, okay, or red color, let us say any color, even you can do this experiment at home, you will see that it mixes quite fast. But if you take the same system and put it 100 ml or 1000 ml and you add 1 drop of dye or red color, you will see that it does not mix as immediately, and that is what, when I say that, this is what exactly represents the mass transfer limitation. So, after adding 1 drop, if you basically shake it up and shake up this as well as this, then the time taken by the dye to mix here substantially decreases. Okay. Of course, the time taken at 10 ml mixing and 100 ml mixing would differ, and the time taken at 10 ml would be this one would be smaller as compared to time taken at within 100 ml. So, this is exactly the what the mass transfer limitation is. Okay. Another important thing that I was talking about is handling of large volumes. This is another aspect of scale up which is a bit challenging. So, handling of large volumes require sometimes specialized equipment. For instance, if you have to carry the media or if you want to carry the media to put it in your reactor and at lab scale, if it is 0.5 liter or 1 liter, you can just carry it by your hand. But let us say the volume is 100 liters or 500 liters, that is 500 kilograms. You cannot carry it with your hands. You would need to basically have either a pump that will pump that media in. Now, if you need that pump, that means that is an extra cost, right. Another thing is that would require essentially specialized pipe which are already autoclaved if you are adding it to an autoclaved reactor or sterilized reactor. If not, you need to essentially sterilize this again. wherever the media or the nutrient is coming in touch with, everything should be sterilized. So, this would be one problem. Another problem, very simple problem, that for taking a sample you can just take a pipette and put your take the sample from the reactor, okay. But in the case of a large reactors, maybe the sampling port itself is 1.5 meter above the ground. You cannot just go there and open it, and the flow rate, the flow that is there would be very high. So, you need specialized valves to actually take the samples. So, all of these things complicate the scaling up process. Okay. So, that is why the scale up itself is a challenge and has to be basically taken up very carefully. Okay. So, one way to do it is that we do pilot plants. Okay. So, what are pilot plants? So, pilot plant is this. This is your lab scale reactor, okay, and this is your industrial scale. Pilot plants are somewhere in between. Okay. They are not at industrial scale, but they are smaller than the industrial scale, sometimes maybe 2 or 3 times, 5 times or 10 times, and at the same time they are 10 times bigger than the lab scale. Okay. So, pilot scale reactor helps us identify parameters that can be used for industrial scale reactors. Okay. So, what are those parameters? I have already described those parameters, but for example, equipment size. What is the size of the fermenter? What will be the size of the bioreactor? Okay. What will be the dimensions of the bioreactor? What is the diameter of the bioreactor? What is the length of the bioreactor? What will be the size of the shaft? What will be the size of the baffles if you have that in the reactor? What will be the size of impellers? What will be the RPM of the impellers? Okay. So, these are the kind of things that pilot plant helps answer this thing. Okay. Then pilot plant help us to identify the other utilities, like for example, we cannot accurately calculate how much amount of water, steam, electricity, fittings and other accessories would be required for a full scale plant. Pilot plant help us to identify and accurately measure these values, or basically accurately estimate these values for the full scale plant. So, these are the things that are pilot plant. When we run the pilot plant, we know exactly how much it is required for the pilot plant, and once we know those things, we can easily extrapolate it, that how much basically those things would be required, like steam, water, electricity, fittings, other accessories for the full scale plant, and then based on that we can do a better cost estimate. We can see that how much amount of things would be required again and again, and what will be the operational cost of running that reactor, what will be the capital expenditure of running that reactor. Okay. And by capital expenditure, I mean what will be the amount that you need to invest to buy those things. Okay. It will also tell you one very important aspect. It will tell you whether it is a labor intensive or how much amount of labor will it require. So, will it require a very highly trained manpower, or a manpower that if you give a training for 3 months can operate it effectively. Okay. So, these are the questions that will be answered by the pilot plant. Okay. And based on that your cost would change. It will also give you enough product to do the market surveys. You can take that product and you can give it in the market for people to see people's acceptance and their or to the agencies, funding agencies, as well as the regulating agencies, to see how basically this product is there and working. Okay. So, essentially running up of a pilot plant gives you production. It will, when you run a pilot plant, what it will give you a techno economic report that can assess the feasibility of the project. So, essentially whether the project is viable technically, whether it is possible to even scale it up, So, these are the things that techno-economic report will give. Another thing is that based on that techno-economic report, you can get financial assistance, for example, loans from the bank to set up the full scale plant. Okay. Many big companies can do it on their own, many other companies take loan from the banks to do it, other companies sell their stakes to do it, and that is how they get the finances to do it. But for doing that, a techno-economic report is essential. Okay. So, pilot plant essentially is a step between laboratory scale production and a full scale production process. Okay. It helps us design and make the path to achieve the industrial scale production, and it not only gives technical details, but it also helps us in getting financial assistance. Okay. So, let us look at an example. So, we want to scale up a recombinant insulin, okay, that is produced at 100 milligram per liter at a cell concentration of 1 gram per liter. Okay. Let us calculate the volume of the reactor needed to produce 1 kg of insulin in the following condition. Okay. So, basically what it says is that 100 milligram per liter of insulin is produced at cell concentration of 1 gram per liter. So, this is what I have copied, 100 milligram per liter and 1 gram per liter of cell concentration. Okay. And what we need to do? We need to see 1 kg of insulin production, okay, under the following condition. Okay. So, what is the first condition? First condition is 1 gram per liter cell concentration, okay, and insulin production is 100 milligram per liter. Okay. So, we know that 100 milligram per liter of insulin is produced, okay. 1 gram per liter of cell concentration is there, and we need to produce 1 kg of insulin. Okay. So, if I write it again, 1 gram per liter of cell concentration, okay, 100 milligram per liter of insulin, okay, in our question 1. Okay. So, what does it mean is, and the amount that we need to produce is basically 1000 gram, that is 1 kg. So, what we need to find out is basically our size of reactor. Okay. Okay. So, amount produced is equal to volume of reactor. So, this is volume of reactor into concentration in the reactor. Okay. So, amount produced is 100 gram or 1000 gram or 1000 to 1000 milligram. This is volume of the reactor and the concentration. So, volume of the reactor in liters, okay, while the concentration that we are talking about is 100 milligram per liter. Okay. So, 100 milligram per liter. Okay. So, let me write this again over here. Amount produced is 1000. So, 1 kilogram equal to 1000 gram equal to 1000 milligram. Okay. This is amount produced needed. Okay. And we know that amount produced is equal to volume into concentration. Volume we need to find out, concentration we know is 100 milligram per liter, volume is in liters, and this is equal to amount produced, which is 1000 milligrams. So, volume is equal to in, so this is V which is in liters, is equal to 10,000 liters. Okay. And the answer is the same. Now, let us see. Now, basically what we know, the second situation is that cell concentration is higher. So, cell concentration is 50 gram per liter and insulin production is per unit of cell is per gram per liter of cell is 100 milligram per liter. Okay. So, and we need to produce, amount that needs to be produced is 1 kg or 1000 grams or 10 raised to power 6 milligrams. Okay. So, I have put the question again here. Cell concentration is 50 grams per liter, insulin production is 100 milligram per liter per gram cell concentration. Okay. And amount is produced is 100,000 1 million milligram or 10 raised to power 6 milligram. Okay. So, we know that amount produced is equal to volume into cell concentration or insulin concentration. Okay. So, if 1 gram per liter produces 100 milligram per liter of insulin, how much will be the 50 gram per liter would produce? Would produce 50 into 100 milligram per liter of insulin. Okay. So, what we need here is 1 10 raised to power 6 milligram of insulin that we need will be equal to 50 into 100 into volume, which is in liters. So, this goes like this goes like this, thousands divided by 5, which is 200. 200 liter of the bioreactor will be needed. Okay. So, how I did it? We know that the cell concentration is 50 gram per liter, which is given. Insulin production is 100 milligram per liter, okay, per gram cell concentration, and amount needed is 1 million milligram or 1 kg or 1000 gram or 10 raised to power 6 milligram. Okay. We know that amount is equal to volume into insulin concentration. We know that 1 gram per liter produces 100 milligram per liter of insulin. 50 gram per liter cell would produce 50 into 100 milligram per liter insulin. So, basically it will be 5000 milligram per liter. So, 10 raised to power 6 equal to 5000 into volume of the reactor. Volume required is 200 liter. Okay. Now, let us look at the third scenario. What is the third scenario? Third scenario is your cell concentration is 50 gram per liter. However, your insulin production has increased. Okay. So, your insulin production is 500 milligram per liter per gram per liter of cell concentration, and how much amount you need to produce, that is the same, okay, which is okay. So, look at again here, cell concentration is 50 gram per liter, insulin production is 500 milligram per liter per gram liter of cell concentration, and amount is 10 raised to power 6 milligram. Okay. So, if 1 gram per liter of cell produces 500 milligram per liter of insulin, how much 50 gram per liter cell would produce? This will be 50 into 500 milligram per liter of insulin, and we know that amount is equal to volume into C. Okay. So, this can be said equal to 50 into 500 into volume. Okay. So, 1000 divided by 25 is equal to volume, which is equal to 40 liters. Okay. So, this basically showed you two things. One is that what see the difference in the, if you have just increased the cell concentration 50 times, basically the reactor volume drops by 50 times. Increasing cell concentration 50 times after a certain level is a challenge, but till a certain level it is not that big a challenge. But decreasing handling 10,000 liter and 200 liter is a very different game. I can tell you, for example, a big container transport can probably carry safely 3,000, 4,000 liters. There are very big transporters as well that can carry 10,000 liters or more, but a small scale small truck can carry 3,000 liters. Now, if you have that truck, basically you can easily manage the transportation and everything with 200 liters, but if you are using a 10,000 liter reactor, that will not be possible with that truck. So, you see that by decreasing the 10,000 liter to 200 liter, you actually make your handling of the liquid much easier, and then decreasing even from 200 liter to 40 liters, it is even much more easier. So, essentially increasing the cell concentration and increasing the insulin production or your bioproduct production from any one of the cells or from the cells is a good way to move forward. And if you do that, basically you can decrease your equipment volume, uh, liquid volume handling, as well as definitely the cost. Okay. So, with this I would like to end this lecture here. In the next lecture we will look at the downstream processing. So, how to separate the product that has already been produced now, and basically how to recover it and make it as a marketable product. Thank you very much for your time, and I look forward to seeing you in the next class.